Okay, thank you very much for this introduction. I'm speaking on behalf of Kelly Tess, Nicole Kmentova, and Maarten Van Oven. I chose to speak because I have the most difficult name to pronounce. They're all parasitologists, and parasitologists are the largest community of users of FishBase, so I also think it's very important that they are at least present in one of the talks. An alternative title for my talk could have been, we found something really strange pattern on gills of fishes, not off, but on gills, and we think we can use the gold to explain it. Maybe that's just because we're biologists and we're a bit theory envy, but if there is an overarching theory, why not try to use it? So, why parasites? So the gold, in a nutshell, relates to the fact that gills are two-dimensional structures that limit life history traits, growth, reproduction, or at least dictate it in three-dimensional water-breathing ectoderms. But on these gills, there are communities of other animals that interfere with gill function. And in this case, these are gill flukes, flatworms, that live on these gills. So for these animals, gills are an environment. And they actually do cause harm on these gills. These are monogenean parasites. They are parasites of fish, frogs, and other water-breathing ectoderms, and also of hippopotamus, but they live on the eyes, not on the gills, of course. They live on skin, but mostly on gills. So the highest density of monogeneans you would find on gills. They're hermaphrodites, so they have both male and female genital organs, and they have a direct life cycle, so they have no intermediate host. They colonize a gill, and they stay on the gill. They're typically very host-specific, so you would expect to find one monogenean parasite species to only infect one species or a set of very closely related species of fishes. They're typically more diverse than the fish host because they also have a much smaller effective population size, a much shorter generation time, so we can use them as biological magnifying glasses. You would expect all patterns or all events that would shape the evolutionary history of the host, the fish, to shape that of the parasite much faster. And that you can use, for example, if you would have an event you can't date because it happened too recently, genetically you would find traces of that in the genome of the parasite much faster than that in the fish. What do they look like? Well, this is one of those worms. I don't think this works, but it's okay. So on the top side you see the head, the prohaptor part, where there's a pharynx and a mouth. On the bottom you see the anchor part, that's called the haptor with an H. The haptor consists of a big anchor and small marginal hooks. On a slide, most you see the same things. On the bottom you see the haptor. That's very specific to the host, so we can see that different species of parasite have different species of haptor that attach to gills of that host. On the top there you see the eyes, it's the other side, so they have eyes on one side and a mouth on the other side, and also the genital organs, so a male and a female organ. Where do they like to live? Well, on gills. Here you see a monogenean flatworm living in between the lamella of a fish gills, where they attach themselves with their hooks, their haptor, that causes the most damage, and where they feed with their proximal part. And they do cause damage. So not only do they affect water flow over the gills, they also cause lesions. This is what the feeding causes. You have deep depressions between the secondary lamella, for example. This can also infect and be then colonized by bacteria. And the hook, of course, causes depressions in the tissue of the gill that can also then infect. Some histopathological slides. You see here a monogenean in red colonizing fish gills, and that can lead to distortion of the cartilage, to rupturing of epithelium, to fusion of lamella, to an increase of epithelium cells, or an increase in mucus cells, an increase in mucus, so all different distortions to the optimal gill functioning. So, what well, we think, and that's actually know, that parasite infections on the gills will reduce gill functioning. So gills will no longer be optimal. So we think that if we find a pattern in the parasites, we can apply the goal to explain it. And it does make sense because what we actually see in nature is that larger-bodied fish tend to have less parasites than intermediate-sized fish of the same species. So it looks like larger-bodied fish have to fight these parasites more actively than intermediate-sized fish. And what is our model system of fish? I haven't talked about fish yet, and I'm halfway through the presentation. It's Lates niloticus. And Lates niloticus is quite interesting because, to the best of our knowledge, it's the largest afrotropical freshwater fish that is dependent on its gills for oxygen uptake. Yeah? So if you find something strange here, well, let's look at gill functioning, let's look at the gold. Lates niloticus is, of course, a very important food fish, and also a notorious biological invasion. So on the left you see the increase of lates catches in Lake Victoria after it was introduced in the 60s, 50s. Yeah? One of the great ecological disasters of our time. Yeah? So lates has a very large distribution in Africa. So this is Lates niloticus and its sister species. So in Lake Tanganyika, they formed an adaptive radiation of 4 species. In some other Great Lakes, there's still question whether they actually speciated or not. So some results point to the fact that they form ecomorphs. Others say they're subspecies or species, but that's another talk. And then the Lates niloticus, the Nile perch itself, you can find all over Western Africa, up to the mouth of the Nile in Cairo and in the Congo Basin, where you have probably a different species, but they look very morphologically the same. Yeah? Normally, you would expect in a species of fish like this, with a very large distribution, with a very large longevity, but very large gill surface, you would expect a high diversity of fish parasites, at least as many as there are species of fishes. You have an adaptive radiation in Tanganyika, you would expect different monogenean parasites on these different species. But that's not what we find. We find all over Africa, there's only a single species of monogenean parasite infecting these Lates niloticus. And interestingly, the sister lineage of Lates niloticus, the sister lineage of the Nile perch, is an Indo-Pacific species living in the Pacific Ocean. And those are infected by several species of parasites that are closely related, so that have speciated within the host. Yeah? And our hypothesis is that it is because, well, this is a freshwater giant that lives in oxygen-poor environments, so the stress on these gills must be enormous, and that might limit diversification of the parasite. Yeah. How would that work? Well, we'd expect in the gills to have stabilizing selection. So you have fluctuating environments in these freshwater tropical systems. So the gills of large Lates perches must be adapted to these fluctuating environments. So they cannot deviate from an optimum too much. And this specific optimum of gill physiology very narrows down the niches available for parasites to speciate on or to specialize on, leaving us with only one species of parasite. We want to test this theory by looking at stress. Unfortunately, Lates is a very poor species as a model system. You cannot put it in a tank in a lab, at least not in our lab. It's also very inconvenient to work with parasites in an aquarium setting, so we have to use collection-based material. And using these collections, we're going to measure the stress on gills and stress on the parasites. How can we measure stress? One thing that's often used is fluctuating asymmetry. So we're going to look at the gills on the left side and the right side of the Lates perches to see whether we find more asymmetry in African freshwaters than in Indo-Pacific marine systems. Yeah? And we're going to do the same for the parasites. Do we find more stress on the haptors, the hard parts of the parasites? Is it more asymmetric in African freshwaters than in the Indo-Pacific marine system? Also, using molecular tools, we can measure stress by calculating the amount of mitochondria per baseline genetic expression. The higher the oxidative stress, you would expect higher mitochondrial counts. Do we find higher mitochondrial counts in African freshwater in the parasite and in the Nile perch than in Indo-Pacific systems? If so, there's more stress, and this could be the cause that makes these parasites fail to diversify. Okay, this is still very much a work in progress. So we're still open for any discussion that can, or any suggestions that could guide this work towards a fruitful end. But, well, I'm here to talk to you the whole day. Thank you.